back together. And Asiyah, do you want to take it away from here since you organized this session? And my gratitude to you for doing so.
Sure, thank you. So I would start with an overview of how we see NCPI use cases taking over the journey of a NCPI use case. Please move to next slide. So I'm using an analogy of from a seedling to a forest. Let's go next. Uh-oh, why is like that? Oh, there are some problems, by the way. Can I share? Somehow it's not showing.
That should be fine, yes. Yes.
Okay, I see in the screen.
Yes, we are.
Yes.
Okay, great. So we will start with what is an NCPI use case. So when we need to answer a scientific question and access, and there's something going on with this slide, but that is general. The wording is a little bit wrong, but it's fine. So access and analyze data from the more than 2 NCPI platforms becomes necessary. So in this case, NCPI use case can be initiated, and then the NCPI use cases need to demonstrate the interoperability. For example, be able to search data sets from +2 NCPI platforms, or access data from at least 2 NCPI platforms to analyze in one workspace, or establish portable softwares and tools across at least 2 NCPI platforms. And there are more examples to showing the NCPI's use cases. But the ultimate goal for the NCPI use cases is to drive the development of NCPI interoperability technology specification. So to make a distinction of the NCPI platforms here, we meant the platforms that support both DDC catalog, CRDC, Kids First, and CBI. So with that, we had looking at a journey. This should be a journey of NCPI use cases that is starting from a proposal phase, goes into implementation phase, and then ended by dissemination phase. During the proposal phase, we mainly focus on produce a one-pager. And then in the implementation phase, it's mainly to carry out the technical plans defined in the one-pager. And then we go demo, training, and reach out to the other users to disseminate to plant a forest from our precious plant. So during the proposal phase, any NIH staff or a researcher can identify a potential scientific use case. After that, we need to collaborate with NCPI working group leads and the platform PIs to try to identify who are the scientific lead and the platform lead. These two are very important. The platform lead can also be an interoperability tech lead. And then develop the interoperability plan and identify the challenges to overcome to achieve the interoperability. And then the finding resources need to be identified in order to implement this interoperability use case. And while all these things being figured out, a one-pager will be developed, and then NIH coordination working group will keep this one-pager for the documentation and the tracking purposes. So this is a one-pager example that is in developing by the Valerie and the Kids First and the NCBS SRA. So they have all the elements identified there, and they're in the developmenting phase. So in the implementation phase, we mainly focus on to make the scientific and the platform leads that they coordinate with the system interoperability working group to carry out those technical plan that is defined in the one-pager. And then they are responsible to report the progress, and they are also responsible to demonstrate at our workshop, and also provide the updates on the GitHub use case tracker. And in this phase, some use cases may become inactive if there's no progress being made. And then when a use case is completed, when the demonstration of the full implemented interoperability technical plan completed, at this point of time, an NCPI use case is completed. There's not necessary to complete the whole research plan for the scientific question answer. So at this point, we start the dissemination phase. And the group who has complete the full implementation, the scientific lead and the platform lead, will work with outreach group to develop the training materials, like the videos that we will put it on the YouTube, or the providing necessary documentations, some blurbs to describe the training materials, the steps that is needed for technical implementation, and any publications that if relevant with the training purposes. And then we use those materials to reach out and educate others to form a forest of NCPI implementation. So this is one example that is already published by the group. Basically, when Jack did his demonstration with all our coordination working group, we just grabbed his video and did some editing, basically by Dave Rogers and his team, and then provided and then uploaded to the YouTube and also uploaded on the NCPI website. So this is one example to show you, and it's not that time consuming because we have already demonstrated. And this is the example of the screenshot of the GitHub site that we created to use to track the use cases. So hopefully after this workshop, people who are creating or proposing the potential use cases can come to this GitHub tracker to add some new issues, and we will track them using ZenHub and this GitHub to just add labels, and we will have a good view of how each use cases is working, progressing. With that, I thank Dave, Jack, Ken, Valerie, and Valentina worked with me to produce these slides, and I welcome you for your questions and suggestions. That is at my end of my talk, so there's no questions or suggestions.
I have no questions or suggestions, but I want to thank Asiyah for trying to put some structure around use cases and processes related to it, because it was badly needed. So I really want to thank you for doing this.
Yeah, I want to mention that this is not perfect. So during our process, actually, we have some new types of use cases developed, like fire use cases. So I really welcome you to give me feedbacks, and we can always change the process to make it more seamless.
Yeah, from an outreach perspective, having the use cases complete and documented is very, very helpful. And you know, the way that they're originally written, the one-pagers, there's quite a bit of you know, abbreviations and so on. So it's difficult to, I think, for someone who's not real close to what we're doing, to parse it. And to have someone then involved in the project be able to expand it out, especially as it's completed, and to show what's actually been done. That would be, I think, very helpful for outreach and getting the word out. We can get the stuff on the site very quickly once we have the materials. So.
Yeah, so Anne was asking a question, is this code that was used to implement in GitHub as well? I think if use case being developed as a material, all the necessary codes, tools will be made available for the users to implement and to experiment.
Okay, why don't we move on? I think our first speaker is Melissa Wilson. But before we start, I just want to remind everyone, the people that are going to be speakers in this session, that you have a total of 10 minutes for your slides and 5 minutes for the questions. So I will nicely nudge you when we are up to the 10 minutes time, and so that we do have time to answer to some of the questions, which is important. So bear with me. The only exception is Valerie and Allison, who merged two presentations into one, so they have a slightly longer time for presentations and questions, but everybody else is 10-minute slides, 5-minute questions. Thank you, Melissa. Oh,
we start with Owen.
We start with Owen. Okay, I'm looking at the.
Oh no, we asked to swap them. Do you mind?
Oh, okay.
Okay.
Pulling up the other slides. It should be in the link. I updated it in the link in the agenda.
Sorry, I'm looking at the wrong. I have not refreshed my page here.
Yeah, no.
So who is presenting?
Melissa Wilson is presenting. We had a last hour swap. If we could go to slide 179.
Thank you, everyone. I really appreciate you swapping, and thank you, Owen. I would be happy to present your slides in some sort of round robin, but I think you would probably do better at them. But I would love to see you present mine also. I think it'd be great. I'm Melissa Wilson. I'm an associate professor at Arizona State University. And I was at the meeting 2 years ago, and I'll just say I'm still just sitting in awe of what everyone is doing and how much is being accomplished, and feel so very small in this room, even on Zoom. So what I'm talking about today is a use case thinking about genetic sex as a biological variable and X-Inactivation. And I wanted to highlight, I think they're not talking about it today, but I think Bob Grossman and Barbara Stranger are doing a really cool project looking at sex differences across some of these data sets. And that's, I think, wildly interesting. But today I'm going to focus on X-Inactivation. Next, please. So as I always start with, I think about language. So I'm mainly interested in genetic sex differences in individuals who typically have 2 X chromosomes and pass on their DNA via egg, and individuals who typically have an X and a Y and pass on DNA via sperm. But as we find out, there's a ton of variation in genetics, in gonads, in gonadal in gender. Next, please. And in particular, these sex differences are multidimensional. So if we think about even XXXY as one component, gonadal hormones is another component, gonad development, time is another component here, where all of these will vary. In fact, we see loss of sex chromosomes over time, so that changes the genetic component, particularly in XY individuals. Additionally, we see that even on an XX individual, you can have something called X-Inactivation. So we'll go to the next slide. And in particular, what X-Inactivation is in individuals with 2 X chromosomes. It doesn't matter if it's 2 X's, 2 X's and a Y, 3 X's. As long as there's at least 2 X's, every X chromosome in excess of 1 will be silenced. And this is a microscopy image here showing one of those inactive X chromosomes as a ball. Those little arrows are showing a ball of heterochromatin in the nucleus. And next, this leads to some observable phenotypes. So calico cats with their patches of orange and black fur, the individuals that are heterozygous for the color allele, which is on the X chromosome, If they have an orange and a black allele, if the black allele is silenced, they'll get an orange patch of fur. If the orange allele is silenced, they'll get a black patch of fur. Next. What's come out though in the last several years is that there's a lot of heterogeneity in that silencing process. So for about a third of X-linked genes, we still actually don't know whether they're silenced or not. There's a lot of reasons for that. Some of it is that humans just have very low genetic diversity, and genetic diversity is what we use to infer X-inactivation. For about a third of these genes, they are inactive in all individuals and all tissues that we've looked at. So we can say that XX and XY individuals will only have expression from one copy of that gene. But for about a third of X-linked genes, they are not inactivated. And we sometimes call them escape genes, but I like to think they're never inactivated in at least some tissues and some individuals. And that's the green bars there, are those genes that have shown escape or never being inactivated in at least some tissues or some individuals over time. And the first, the dark green column is all genes that have shown some evidence of escape. And then the next two columns are looking at immune-related genes, about 50 some odd of those, and then tumor suppressor genes or tumor-related genes that have been shown to escape inactivation in XX individuals, but there's no capacity for that in individuals with a single X chromosome. Next. So one of the things in thinking about use cases that we're interested in is X-inactivation broadly, and I want to show you a little bit of data motivating this from the human placenta. So the placenta is largely the genotype of the offspring, not of the pregnant person. And so that means that you can have really unique interactions between the placenta and the pregnant body at that interface due to the genetic component of the placenta. And so in this project, we're interested in looking at patterns of X-inactivation across the placenta. Next, please. So we were aiming to first distinguish between these three scenarios. So we look at two different regions of the placenta, and we compare those to try to see if we can infer whether there's complete skewing, so you only observe maternal or paternal X chromosome, whether there's large patches of maternal or paternal X chromosome, or whether it's completely mosaic on the right. So the three in the middle are three possible scenarios for patchiness where we might observe only mom's X chromosome, moms and dads, or a little bit of both because we've taken a chunk of sample at the boundary. And the last scenario is that it's mosaic, that we observe both maternal and paternal X chromosomes being expressed, but because those cells have kind of migrated and moved about, we observe any bulk sample having both alleles expressed. Next, please. And to skip all of the details because we're short on time, I'll say that using RNA-seq and DNA-seq across 30 individuals with 2 replicates per individual, we very concretely believe that the placenta is patchy in nature and that there's large patches of the placenta that express only maternal X and large patches that only express paternal X chromosome. And so this is really curious if we start to think about some diseases of placenta in development that show a large sex difference and whether this patchiness is contributing to protection or not. Next, please. And then a big question is the placenta distinct from adult tissues? And so we were able to compare against the GTEx consortium data where there was a tissue, in this case, the heart that had left atrial appendage and left ventricle. And spoiler alert, it's very different. In fact, so the placenta, we see these large patches of maternal or paternal X being completely inactivated. But in the heart, in the left atrial appendage and then the left ventricle, we observe mosaic X inactivation across a lot of them, almost all of those samples with very little skewing or very little patchiness. Next, please. So this leads to a lot of other questions. So a second part that I'm not going into is we're also looking at, once we know whether it's maternal or paternal X that's inactivated, which are the genes? So I told you some genes, about a third of them are going to escape in at least some individuals or some tissues. So we're looking at in the placenta, which are the individual genes that are escaping across regions of the placenta. And we want to see, are these genes unique to a tissue, unique to a particular condition? For example, there's been some really great wet lab work done showing that some genes escape only in T cells and B cells. So they're escaping only in immune-related cells and do lead to differences in immune-related function in T cells and B cells. But we don't know, right? What are these patterns of X-inactivation broadly across cancers? Are the individual genes that are escaping the same or different? Are they contributing to some of the sex difference that we observe across cancers? Are they the same in pediatric and adult samples? We don't know. It's just kind of a big world of we don't know. Next, please. And then that leads me to what I talked about a couple years ago, is that genetically and genomically, when we're doing the analysis, there's a big problem with the sex chromosomes. And that's because they share an evolutionary history. About 150 million years ago, the X and the Y were identical. They were homologous chromosomes, and we couldn't distinguish between them. And they today, although they've distinguished a lot from each other, they still retain a lot of that homology. Next, please. And so what happens is, following standard genomic mapping pipelines, we see that individuals with 2X chromosomes, some of their reads will map to the Y chromosome. And so if you pull down Y-linked BAMs following standard alignment procedures, you'll find reads aligning to the Y chromosome in every sample in your set, even though hopefully you're following NIH procedures and including sex as a biological variable, and at a minimum including both males and females. You'll still see that the Y is in every single sample. And that's scary, because those Y-linked reads shouldn't be there. They're wrong, and they're reducing our power to detect gene expression and identify variants on the X chromosome. And so one of the things we want to do is to remap everything. And that is, in thinking about what Michael said and other people have talked about, it is just a mind-blowingly large amount of work. And how do I even estimate costs for doing that? And Brian and Becky have been great about showing that it's feasible, but me being this little fish in my little pond at ASU is like, really? How, right? We've had these ideas, but how? Anyway, and I sat in 2 years ago, and I know better than most, and I still don't know how. Okay, next, please. So we've come up with a software, XY Align, to appropriately infer sex chromosome complement, map the samples and realign. Next, please. And I'm almost at my time limit. And what I just want to show you is this big take home, that this mapping really matters. I can show you it in a lot of different ways, but I want to show you this is sex differences in expression. And these are individual genes that I've pulled out when you map standardly on the top versus sex chromosome complement on the bottom. So the first is that you do our mapping and you say, Melissa, it doesn't matter. Look, they're differently expressed in both. And I'll say next. And I'll say sometimes we go from observing no sex difference to now observing a significant sex difference in an individual gene. Next. Sometimes it's not different in both. Look, why'd you make us do this? Why'd you make us remap? It's not different anyway. Next. Another gene where we go from being sex differently expressed to showing no sex differences in expression when you do appropriate mapping. And finally, and this one kind of blew my mind, and I just randomly pulled these genes of my favorite genes of interest. We go from having a male bias to a female bias when you appropriately map. So yes, it's a small number of genes, but it's going to have a large effect on our results. Next. So with that, the thinking about where we're going to go and opening it up for questions, in studying X-inactivation and X-linked genes, I think it matters for everything. We know that there's a male bias in most adult cancers, in most pediatric cancers, in heart disease, in susceptibility to COVID-19. The ACE2 receptor in particular is on the X chromosome. Next. And finally, so heart disease, I did mention, is more biased in men, but actually after menopause, females have a significant upward trajectory in heart disease, significant bias in autoimmune disease. And although less susceptible to COVID-19, account for more than 90% of the adverse reactions to the vaccine. And so there has to be some underlying genetic component here that I think is really worth looking at. Next. And so I want to thank the people in my lab, funding from NIGMS for this. And last, next, please. I want to thank James Taylor, who was here 2 years ago, and he really got me involved. And Brian and Becky for keeping me involved. And this last bit that I think is really important, that good ideas don't have owners. They belong to everyone. Thanks.
Thank you, Melissa. We have 3 minutes for questions.
I have a couple of kind of like.
Stanley?
Oh, I'm sorry.
Stanley?
No, let Mike.
Mike.
Sure. I have a couple kind of little comments. So one is, before doing too much, I would wait maybe like 2 weeks, because we're really close to getting a T to T chromosome Y that I think will further improve the mapping. And
that's super exciting, because I was waiting for that. They were like, we got the telomere to telomere of the genome, and I was like, almost.
So there's a draft now. It's in final polishing, so that'll be available soon. I'll make sure that's available in the Anvil workspace. And then kind of a more broader question is, I would imagine like single cell data would also be extremely valuable for looking at mosaicism. Are there such data within the NCPI universe that would add further power?
I don't know. I would ask the people who know the data better.
Mike, we have a cohort entirely on disorders of sex development, so you can sort of look at different combinations of X and Y chromosomes. It might be interesting for that.
I think that's a great idea. I think the DSDs are really important for really disentangling the hormonal from the genetic component. Not completely, but they're a natural occurring system. So I think that's really important. I didn't know that was here.
How about the Include project, Valerie, with the Down syndrome samples?
Yeah, that would also be interesting.
Stanley, do you still want to ask a question?
So I just wanted to say that I really support calling the X chromosomes. I think this is a great idea that we heard 2 years ago, and I'm so pleased to see it move forward. And this captured a lot of people's attention, and I'm glad that you've gotten the support. And I'm just pleased, and it's wonderful to be involved in something like this.
I just going to also
not a question.
I was also going to advocate, from the Kids First side, that there are cancer cohorts that are there. And to your point, I think this has been long noticed, right? This differential in the predisposition, hypothetically determined by germline settings of sex, even in that setting. And then, for many of those, for some of those cancer cohorts, there's expression data, and then furthermore, proteomic data as well. So you could correlate even some of those differences.
I would love to pick everyone's brain about their ideas with this, and we could put it into one master project that is like stage 1, stage 2, stage 3 of I don't know. It'd be great.
Thank you. We need to move on. The next speakers are Valerie and Allison.
Awesome. Thank you, Melissa. It's hard to follow you every time. It's always a good talk. So Allison and I together are going to do this presentation. This is a new use case, and we're presenting on behalf of a lot of people, just to set the stage for what we think is going to be a really interesting, exciting use case and collaboration. Next slide. So this is a new use case, and broadly, the goal here is to enable researchers to easily co-analyze data from Kids First and the Undiagnosed Disease Network in the cloud to leverage the large-scale pediatric cohorts that we have in Kids First to resolve variants of unknown significance from the UDN cases. This brings together 2 different common fund programs, and you can see pretty clearly where these goals intersect. Kids First, of course, the goal is to uncover the genetic etiology of childhood cancers and structural birth defects. And the goal of the UDN is to improve the diagnosis of rare and undiagnosed conditions using both basic and clinical research methods. Next slide. I just want to briefly show, this is from, on the right, the graphic is from a recent report from the UDN, but it's actually, this report was published before the recent release of data, but you can see that there's actually a pretty heavy emphasis on the UDN, on pediatrics in the UDN cohort, especially for the genomic data they have. But as of last week, the SRA has genomic data from more than 4,000 participants from the UDN. Next slide. Okay, and this is Lisa. I'm not sure if she could make it today, but because we have such a short amount of time, I am going to try to explain her work on her behalf very briefly. So she is part of the UDN site at Vanderbilt, and she has created this pipeline to generate phenotypic risk scores, which indicate the level of overlap between UDN participants, and she does this comparing with participants that are part of the Vanderbilt medical system. She queries the EHRs for variants at Vanderbilt, and then when she finds participants who have variant matches with her UDN cohort, she assesses that phenotypic risk score from her algorithm, and then uses that information to try to resolve variants of unknown significance. So in this use case, we are going to try to support her in applying this method on a much larger scale by using the childhood cancer and structural birth defects data from Kids First to try to resolve those variants of unknown significance in that larger, 4,000 plus UDN cohort. Next slide. This is a high-level technical overview of the standards that we're planning to test and use to support this use case. Primarily DIRS for enabling a user to pull those genomic data files from multiple sources into Kavatica. And FIRE will serve the same purpose for this phenotypic data. And I think Robert's talked about how DIRS and RAS are kind of serving a similar function here. They're both agreed upon approaches for serving up different types of data in a standard way. They're both used by NCPI, and of course we use the same standards in Kids First and across NCPI. So it just seemed like it was time to test this out. RAS, of course, permeates all of the systems here. And here we're actually going to use it for authorization and authentication to get the UDN data from dbGaP's DIRS server. And we're also using HPO and CWL. So this use case is just totally rife with all the standards. Next slide. So there's multiple layers and components to solving, to offer solutions to get at this use case. There is a genomic data flow and there's a phenotypic data flow, and there's work from the Kids First side and work from the NCBI and NLM side. And of course, our researchers who are actually going to run these analyses in the cloud. I'm going to talk about the genomic part, and then Allison will talk about the phenotypic part. But briefly, we know that Kavatica already knows how to talk to the Kids First Gen 3 DIRS server. Here, they will connect to dbGaP's DIRS server, and this requires using RAS v1.1. Typically, SRA BAMs actually live in cold storage, but to support the use case, we needed the files to be accessible to DIRS. So they actually needed to be in S3. And as long as the data reside in AWS US East 1, in a repository that is that AWS region, and the workspace that we use is also in AWS US East 1, there will be no egress to access the data, and DIRS will work as it should. And then I'll talk a little bit more about the downstream analysis as best I can a little bit later. So next slide. And of course, this is just going to take a all hands on deck situation to really tackle this use case from many angles. So we sort of assembled this team. We've gotten started on some of the other parts, but following the workshop, we will dig more into the technical implementation. Next slide. Okay, these slides are from Michele. So, this just shows you that a key goal here is to use RAS and DIRS as they are intended to be used. So in order to do this, 7 Bridges will have to obtain that RAS v1.1 passport directly from RAS and present it to the dbGaP DIRS server. And of course, we all confirmed in this process that they have to present the entire passport and not just the visas that come from RAS. And the outcome is the signed URL from or URLs from DIRS that enable the user to access the data in the workspace. Next slide. The user is going to have to connect to that DIRS endpoint, that new endpoint that is from NCPI within 7 Bridges. So the 7 Bridges team is just proposing to add this card to make that easy for the user to connect. Next slide. Okay, and so in order to access DIRS, you have to first obtain the SRA runs that you're interested in from the SRA run selector. The SRA run selector does not store the DIRS IDs, and hopefully Kurt is still on the line, so he can chime in if I'm saying something wrong. But you have to use this NCPI indexing service to basically transform those SRA runs into DIRS. The first time you call DERS, you actually get a bundle for each SRA run. And this is because often one SRA run has multiple files, like let's say BAMs and FASTQs. And then you need to, call again with the actual DERS IDs you're interested in. But Seven Bridges will have to design an approach for unpacking that bundle to select those IDs and then to access the associated URLs for the specific data location of those files. And again, here we're taking the AWS route. Next slide. This is just a draft proposal, but ideally a user would not have to go, through this process of SRA run selector or indexing service. So Seven Bridges is looking into maybe building the DERS links into a public project in Kvatica. So once a user logs in through RAS as this public project, they still need to get dbGaP approval. And RAS will carry those dbGaP permissions, but the user will more easily be able to analyze the data in Kvatica because those DERS links will be built in. Next slide. Okay, and I probably, again, am representing a lot of people. So on a high level, there's multiple ways to actually do that comparison of variants and phenotypes in between the UDN and the Kids First data set. Here, we're proposing to call the UDN variants based on those bands with Kids First workflows so that we have functional equivalence with the Kids First data, to identify the variants of interest that you're then going to try to solve with the Kids First data set. But the user could actually use the Kids First variant search function in our portal to find which Kids First data set she needs to apply to. So she doesn't actually have to apply to every single dbGaP study in dbGaP. She can search on the individual variants or genes, find the relevant dbGaP studies, apply to access those so that then she can assess the row level phenotype genotype match of like what phenotypes are associated with those variants to do her phenotype risk score analysis. And the next slide is for Allison.
Great, thanks, Valerie. As Valerie mentioned, we're representing a lot of people across this, but really trying to be practical and driving it forward with, the specific phenotype risk score pipeline. I'd say, as we're going through the kind of solution matrix, really thinking about how this will be a very practical application of can we really do every step that it takes to do this along the way and really force ourselves to examine all the steps and say, is this the right way to do it? So if you go to the next slide, I think, there's a lot of good substrate here for us to work off of, especially because this pipeline, I think, is representative of a lot of ones that I've heard about. And since, I think Lisa is really willing to be engaged and help with this, a lot of these kind of clinical informatics tools are R-based. It's a very different world than the genomic pipeline world of large scale algorithms running for hours and days. They're often, leveraging in-house databases and then having different kinds of statistical analysis tools that are being run over them. And how you take something that might be customized to a local infrastructure with a local database and put that into a cloud-based platform such that it can be run against other kinds of data types across data that might be in SRA or Kids First is really what we're looking at here. And being able to do that to a new cohort. So a lot of times these are developed, from a specific institutional or study perspective. Well, we have the opportunity here with these interoperable resources are to create these virtual cohorts and to be able to intersect that data, as well as making it more available to the community and start thinking about how do you have the utilization and comparisons to other kinds of work, just like we do in the genomic space. Go to the next slide. So when I heard about this use case, I was really excited because if people aren't familiar with UDN, I think I was at one of their meetings 4 or 5 years ago, and it was like, wow, this really is like House MD happening in real time with a bunch of people who know a lot about rare diseases, going through patient by patient, looking at the phenotypes, looking at the variants. And I had, inkling then, I think a lot of people did of, okay, this is pretty cool to be able to get together and do this kind of one by patient, but we're both creating a lot of information that can be used to understand other diseases in the rare space, as well as how would you bring in information from the larger landscape to help inform what's going on with the UDN. And so over time, a lot of different methods have been developed, such as the one we're talking about today, but really also thinking about, okay, we have HPO, we have variants on the UDN side. Kids First also has a lot of different phenotypes, rare diseases, different things across the pediatric space. So here we have phenotypes and variants from UDN. If you go to the next slide, on Kids First, we have all these phenotypes, and then I picked actually just one of those variants from the previous one there and looked it up in our portal's variant search. And you can see there's 18 participants on the Kids First side, where if you're in the space of the rare, actually that's pretty great to be able to find 18 participants with a variant that you want to look at, and then you can incorporate the different phenotypic data. And so we have all the substrate here. How do we actually connect the two, not just from the genomic data perspective, which we've done a lot in the Kids First side, but really also start pushing it on the phenotypic side, and then ultimately intersecting the two. So if you go to the next slide, I took Robert's slide from yesterday to really think about, okay, to make this happen, which steps do we really need to take? So he had presented this slide here of the different services, a general idea of a phenotype translation tool and a general idea of a research user. And so what we can do here is make those boxes a little more concrete. So you go to the next slide. Here we're doing Kids First and dbGaP. This is the first two pairwise ones. Obviously, we would like to see this expand across the different ones, but just from a, let's get started standpoint. And then we need a translation tool that will take that data out of FHIR, find the HPO terms, and translate them into a list or table that can actually be fed into this phenotypic risk score pipeline. So we'll have to figure out what that is, how general, how specific do we want to make it specifically for this pipeline, what are the trade-offs there, going into the phenotype risk score pipeline, and then ultimately genomic and phenotypic pipeline results. And so I think each one of these steps, if we go to the next slide, I'll start to talk to that first one of, which protocol is the most practical and useful. We've been talking a lot about PFB. FHIR bulk has ND-JSON. There's the new emerging landscape of using more generic Avro for FHIR. Is it something else? Is it none of these? Is it really just, you can actually pull from the API directly into R, and that's perfectly fine. As Valerie alluded to, I think this will push on the RAS-based access to the FHIR data, and how do we do that? And to get from this step to this step, those are the questions we're going to have to answer. Go to the next one. Then once you have that data, it's transformed, it's come out of the FHIR server, it's in a form that can be input into the pipeline. We've talked a lot about Docker files and other things for the genomic space, but what kind of tooling do we really need for the phenotypic space? Do we need to be able to deploy on the fly database? Do we need to support other kinds of capabilities? Our studio, our Shiny, what goes on here? Is it Dockerized? Is it something that can be packaged up and just automatically run? So really kind of taking that tooling out from the local infrastructure and saying, what does it really look like in the cloud workspace? Kind of with the goal that you can go more directly from a virtual cohort to an automated pipeline that will then generate those results for all the different participants you're interested in. And I think there's a lot of questions that we'll hopefully address and answer here. And then the last one is, once you do have that, how the tooling goes in the cloud workspace. If you go to the next slide, please. I think that my hypothesis is this is probably the most well-defined, that once you have everything kind of in tables across both the phenotypic and genomic space, this is where a lot of the current features and the platforms are addressed to. You bring up your RStudio, Jupyter Notebook environment, and kind of do your scientific analysis. So I'm hoping that by the time we get to this step, we're pretty good to go. But you always get surprises when you try to go through these use cases for real. And really, to be able to answer that scientific question, do those variant matched individuals have unexpected elevated phenotypic risk scores, and to really, start digging into and understanding the scientific part there. And so I think you could legitimately say that's probably the practical end-to-end use case that we need to do. I think I would throw out one last thing about this, just from what I've seen in terms of people doing these analyses, finding interesting things, especially in the rare, and saying, wait, is there more information available? What was the source? This kind of looks right, but I'm not sure if that HPO term is really the right one to be associated with this variant. Did they have some other kind of syndrome, other things going on? And so in the last slide here, I think the hope is that with the FHIR services and being able to have it available there, it's not just about getting to that end to end, but once you do find something interesting scientifically, you're able to go back, get more data, more information, go back to the source, And I think talk about this world of, can you pull in more data from EHRs, other things, you know, especially when you're looking at these kind of case-by-case basis where you're trying to really solve what's going on the UDN side or on the Kids First side, where you're really trying to understand and discover, you know, what's going on with these different variants in the developmental landscape. And some of the discussion we've had is it's great to start with HPO. That's a good place, but, you know, are there other things we can pull in, like the ICD codes that can actually feed into these type of pipelines as well? So hopefully me and Valerie got through that in time, and happy to take any questions. As we both said, there's a lot of people involved here, so different people might be able to answer different aspects of it.
Thank you. We have 3 minutes for questions left. Ben?
Hi, Allison. Thank you all for the presentation, and it's super exciting. Great work. I'm very, very happy and impressed by the progress. I have one sort of out of left field question, because that's what I do. And that's, I'm curious, Allison, in your presentation of end-to-end state, do you think that getting the data in some sort of data frame structure and into a Jupyter notebook or RStudio environment for a bioinformatician to work with is really the ideal end state? Or is that sort of the end state for now? And I might suggest there might be other things that we can envision in the future that would make analysis a little easier or more cloud native or reproducible? Or do you think, is that the end state in the end to end?
It's interesting, because I think the mathematician in me says, what can't you do with matrices in some sense? and that's I think that's why you always end up in tables in some sense to do analysis. But I think you're right in terms of, can you mint them as this object, right? Like, this is the kind of table or matrix that I got at this time for my analysis, especially given that, a lot of these things are updated longitudinally or changed. Is that what you're kind of going with? Like, how would you reproduce this and kind of have maybe a data object around your clinical data, not just lost in someone's notebook or our code? Is that, I understand what you're asking.
I see Paul chomping at the bit, so I'll let him.
Okay, yeah. Paul, and I know, Paul, you've been heavily involved in UDN over the years, so.
Paul, we lost you.
Lost you.
Well, Paul's coming back, maybe one other comment, we've talked about this a bit in some other presentations, but one of the, as Allison mentioned, this House MD work, they're not using Jupyter notebooks. And so, the other next step that we really are interested in is, democratizing the capacity to search across this space. You have this dedicated work here with a specific researcher, but this work itself will create additional opportunities to expand the searchability across these cohorts in ways that we can further empower, for the next House MD setting.
Yeah, maybe the feedback loop I don't really have here is that scientific analysis to portals and searchability is what you're kind of talking about, Adam, of like, okay, Lisa or some other person is going to do all this great work, find all these phenotypic FISH scores, bring new knowledge to understanding this. How does the clinician with the new variant of unknown significance be able to find out that, okay, actually there was a case or two found in Kids First or UDN or other places, even if it is in the research space, to kind of more holistically inform what they're thinking of.
I apologize, but I think that we need to move on. I'm not sure what happened to Paul. Eventually we'll be back. Okay, let's move on to Owen.
Is it okay if I share my screen? Just, there's a lot of animation. Thank you. Okay. Hello, I'm Owen Hirschi, a graduate student in Dr. Sharon Plon's lab at Baylor College of Medicine. And I want to thank the NCPI for allowing me to update everyone today about the progress of a new collaborative project between our lab, Dr. Michael Schatz's group, and the team over at Seven Bridges Genomics. So to give a brief intro into the project, our lab studies pediatric cancer predisposition through sequencing data. One cohort we use in the basic 3 childhood is the basic 3 childhood cancer cohort that was originally a clinical germline matched tumor whole exome sequencing study, analyzed here locally at Baylor and then stored on dbGaP. Then, through Kids First, we performed germline trio short read whole genome sequencing on a subset of these samples. That data is currently being housed and analyzed on the Kids First Cavatica platform. Now, in a follow-up study to Basic 3, the Kids Can't Seek cohort is currently being analyzed and stored on the Terra Anvil platform. This is all to say that our lab, like many others, utilizes multiple platforms for storage and analysis. Recently, through Kids First, the Basic 3 cohort was selected to undergo PacBio CCS hi-fi long read sequencing. Our lab in particular was interested in this technology given the increased ability to detect structural variants when compared to that of short read data. We plan to leverage this trio data by prioritizing the identification of de novo variants not occurring in the healthy population, and we'll do so using 2 major algorithms, the Minimap2 aligner and Sniffles structural variant caller, currently available on the Cavatica platform. As I'm sure everyone here is aware, the complete sequence of the CHM13 human cell line through the telomere to telomere consortium was just completed, which through a combination of these long read sequencing technologies and analysis on the Terra platform introduced nearly 200 million base pairs of novel sequence to the reference genome. However, one of the problems with analysis on platforms like Cavatica and Terra is that tools are written and uploaded in using different workflow languages on each platform. Specifically, on Cavatica, tools are written in the common workflow languages, while tools on Terra are written in the workflow description language. This may not be problematic for some who use one platform for their data, storage, and analysis, but there exists labs like ours who have data on both platforms and would like to perform identical analysis on both, but need to ensure the equivalency of these tools uploaded on these platforms in their respective languages. So for example, tools like BWA, MEM, and Picard both exist on each platform, but may differ in their version and the resulting output using the same input and the same parameters. Along with this and the continually increasing use of long read sequencing technologies, we wanted to test the functional equivalence of long read sequence aligners and structural variant callers on both the Cavatica and Terra platform. For this cross-platform analysis, we wanted to use a well-established benchmark set who had long read sequencing technology performed. In this case, it was the well-documented HG002 trio from the Genome in a Bottle consortium. This trio in particular has had multiple technologies applied to it, including 3 different long read sequencing technologies from PacBio and Oxford Nanopore. And for this specific test, we prioritize the analysis of hi-fi CCS data, given it's the type of data we'll be analyzing with the Basic 3 cohort. We also wanted to use a benchmarking pipeline that was accessible on both platforms and similar to our future analysis, including the Minimap2 for sequence alignments, Sniffles for structural variant callers, and Survivor for variant merging. The latter of these 2 algorithms were developed here at Baylor. And in order to generate this pipeline on Cavatica, I utilized the public applications aspect of the platform, which utilizes these common workflow language apps to run with specific parameters. Then, utilizing the data cruncher feature, we can run Survivor, which is a non-language dependent variant merging algorithm. And then finally, we use IGV to merge these visualized variants, or to visualize these merged variants. And then for the data generated on Terra Anvil, we relied on data previously generated by the Schatz group on the HG002 trio, using Minimap2 and Sniffles as a part of one of the telomere-telomere studies, and a novel structural variant comparison tool called Jasmine, both of which are available to read on bioRxiv. Given this project is still in its very nascent stages, we do have some preliminary results on the data generated via both platforms, but I hope to have more in the coming months. So when we look at the alignment data, we notice a small but consistent decrease in coverage across all 3 samples on Cavatica when compared to that of Terra. Then when we look at raw structural variant counts, we again see roughly similar counts between the 2, with Cavatica consistently calling 4,000 more unfiltered structural variants per member of the trio. Finally, when looking at disconcordant variants, or those found only in each sample, Cavatica again sees between 4,000 to 1,000 more variants per sample. And I would again like to emphasize this is using the same input FASTQ files, same reference genome, along with similar, if not perfectly identical parameters for each of these tools. And while I haven't yet been able to dig into the reasons for a lot of these differences, we have been able to look at some of the potentially discordant / de novo variants only found in the proband to see if this was similar across both platforms. So the first example is 107 base pair deletion only found in the proband on chromosome 17, the Schatz group identified, and that we were also able to re-identify in our variant calls on Cavatica. The second is an example of a 34 base pair deletion only found in the proband on chromosome 18 that the Schatz group also identified in their previous analysis, and we were able to re-identify. This is to say that while we do have count differences between the two platforms, there were a large number of shared de novo events we were able to resubstantiate, and for most variants, we were able to call the exact same breakpoint and length of variant. So in summary, long-read sequencing analysis tools are uploaded on these platforms in different coding languages. We have been able to set up a functional long-read sequencing analysis pipeline on Cavatica. We've also been able to identify these de novo variants previously found via pipelines on the Terra platform, but we've also identified a roughly 5 to 10% difference in both raw and merged structural variants across these two platforms. And in our ongoing work, we're trying to understand the differences in these called de novo events and aligned sequence files in this trio on both platforms. And we'll do so using the new Jasmine program that allows us to both merge and filter these variants to see if post-filtration, we get a similar number of variants. And then the larger goal is to determine if there's a larger data set beyond this one trio that we can process on Cavatica and Terra respectively to test the full functional equivalence of these tools. And finally, we will eventually perform long-read sequencing analysis on this basic 3 cohort using this pipeline we've set up to identify novel de novo structural variants. And with that, I want to thank everyone in my lab, the Schatz group, and those over at Seven Bridges for their continued help and support. And with that, I'll take any questions.
Oh, and thank you. This has been great. We have plenty of time for questions, and I see Anne raising her hand.
Hi, Owen. Wonderful work. So I hope to connect with you, but just a really simple question. So workflows should be, it's a language that stitches things together. So are we certain that they're using containers that house the same environments and versions of software? Because that's one way you can normalize across languages.
So we've definitely normalized version-wise. Docker site-wise, I think that's a bigger question that hopefully I can get the answer to, at least on the Terra side. But yeah, version-wise, they're perfectly identical.
So you start with the same conda environment before you build the Docker and everything?
So the building of the actual tools is not done by me. These are tools that have been already uploaded onto Seven Bridges and processed onto Terra.
So one of the things that, you know, I'm a big fan of is automatic building of the Docker images, which Docker Store doesn't allow anymore. I don't know if GitHub does now with its container registry, but those are like, that would just be where I'd start.
Jack.
Yeah, so, oh, sorry.
Obviously, wonderful presentation, Owen, and sorry, Brian, for jumping over you. Can you comment also on the random component of, I think it was sniffles that may be contributing to this?
So I can't comment too much on that, just given this is my real first experience with sniffles as a structural variant caller. But I think Dr. Schatz is a better person. I know he just left to sort of talk about how that tool does input some randomness. And it's either sniffles or Minimap to input some randomness in either the alignment or the actual calling. And that could contribute to some part of this.
Brian?
Yeah, so Owen, I was just trying to understand, like, and this is kind of getting at Anne and others' questions here. Maybe I'm misunderstanding the sort of punchline here, but it sounds like all the tools are the same version, but you're seeing platform differences, and you're not comparing functionally equivalent pipelines. You're comparing the same tools just run in different places. Am I understanding the punchline here, or did I get that wrong?
No, that sounds fairly close to the punchline.
But I just argue a little bit. I argue a little bit. I think that's not true, because you don't know how the Docker images are built, and you don't know, you know the image is there, or are they pulling from the same image?
Yeah, I think they're definitely pulling from different images. I think the random component is something that we have to take a closer look at, and unless you actually set the random seed exactly the same, we would expect some differences. I guess the question is, as you get to a larger database, a larger data set, would that average out or not? But I really like this for like the inverting the interop paradigm a little bit and looking at the things potentially coming out of recover and PASC where the data can't move and we have to send the compute around, and what does that look like? I think that's really neat.
Yeah, Jack, and I just want to underscore, like, I think this is kind of highlighting as we move algorithms to where the data lives, as you're saying, what are the challenges with that? So if you're operating with the same tool, you kind of make this assumption of like, oh, it's the same tool, it's the same version. But this is showing that there are some, whether it's random seed, whether it's the platform architecture, the actual CPU and the libraries that were used, as Anne was saying, you haven't controlled necessarily the compiled environment, the Docker container that's running this. So it's definitely underscores like lessons learned in terms of interoperability and the importance of why it's really important to move algorithms around and actually check your assumptions that they're going to produce the same results, because there's a ton of dependencies within a Docker container, system libraries, version of the kernel, underlying platform that may throw off ever so slightly answers and produce something like this. So I don't know what the answer is as to why the two platforms are performing differently, but it's really important that we go through a process like this as we're moving our algorithms around. And I'll be fascinated to find out really what's the underlying difference here. Is it a library issue? Is it an architecture issue? Is it a Docker version issue? Like, what really is throwing off the numbers and something that should be consistent?
Michele?
Thank you. Hey, hi, Owen. I have a question. Did you try to run exactly the same samples with the same pipeline on the same platform and verify if there is a difference in the variance over there that you get?
So no, we've only had this data for about 2 weeks in terms of just running it on the same platform multiple times. I think, is that what you're asking?
Exactly.
Yeah, no, we haven't gotten a chance to do that, but that is something to look at. I think part of this project was also sort of setting up functionally doing this on Kavatica just in the first place.
I think it will be extremely worth doing it, especially for the stochastic nature that some of these tools brings in, and you might see this maybe even in different times and so forth, to see if there is a variation over there, because it might influence as well. So thank you.
Adam.
I just want to make a comment that that Owen, I think you also inserted as part of your slides, which is, and we see this when we do some of these benchmarking and comparisons, where oftentimes, you might have 5 or 10% differences, but much smaller percent differences on the things that someone would have moved forward on, post filtration and or at significance levels. So I just wanted to, I think you were sort of pointing to that, and we've seen this a number of times. And I think that's an important narrative, for us to communicate to the community, because people are always worried about missing things, but sometimes you're actually not as much. I don't know the perfect answer to that, but we've seen this.
I mean, Adam, I agree, but that's one of the reasons that Owen also did the de novos, right? Because almost anything de novo is going to get moved forward, and the difference is smaller, but there's still a difference. And so I think, he's only, as he said, had a short period of time to do this, but I think really digging into the differences in the de novo calls, which those are calls that almost any platform would move forward, would be particularly informative.
Oh, no, yeah, I wasn't trying to minimize the worthwhile nature of this. You know, sometimes people just get scared when they see like large differences. And so to your point, like those differences also get smaller and potentially more tractable as you start honing in on like you said, de novos.
I think we need to move on to the last speaker, Simran. Thank you.
Hi, everyone. I think Paul is going to be sharing the screen for this. Paul, do you already have the screen shared?
I see it. Do you see it?
Perfect. So hi, everyone. My name is Simran. I'm working in Paul Avillach's lab here at Harvard Medical School, and I'm part of the PIC-SURE team here with Biodata Catalyst. Next slide, please. Thank you. I'm going to explain a little bit about what PIC-SURE does so that you can understand how it was useful for the two scientific use cases that I'll be presenting today. The first one being PIC-SURE working with 7 bridges to do ORCID study reproducibility, and the second one being PIC-SURE and Terra for HCT for SCD. And then I'll pass it back over to Paul to talk about PIC-SURE as a search tool across NCPI platforms. Next slide, please. So PIC-SURE stands for Patient-Centered Information Commons, Standard Unification of Research Elements. And users are able to utilize PIC-SURE using either the user interface or the UI or the application programming interface or API. The user interface allows users to search available data at the variable level using queries to build cohorts using a simple point-and-click interface. Results can then be exported via the API for analysis. Additionally, data is searchable and queryable through the API, and the API allows for data to be exchanged between PIC-SURE and other platforms. While PIC-SURE is used for many different projects, the use cases that we're discussing today use PIC-SURE powered by Biodata Catalyst. And PIC-SURE powered in Biodata Catalyst currently integrates clinical and genomic data sets across, including TopMed and TopMed related studies, COVID-19 studies, and BioLink data. Next slide, please. PIC-SURE provides access to data in 2 different ways, open access and authorized access. PIC-SURE Open Access is available for anyone with an ERA Commons ID to log in and explore aggregate counts of phenotypic data sets. So anyone on this call right now with an ERA Commons ID could log into PIC-SURE while I'm presenting and explore data with PIC-SURE Open Access. For those of you who do have access to data with dbGaP approval, PIC-SURE Authorized Access is available and provides users with access to all variables. So this is clinical and genomic variables at the participant level. Next slide, please. So to speak a little bit about how PIC-SURE improves the user experience, I'll walk through a brief explanation of how data stored in dbGaP is traditionally stored and accessed. Clinical data in dbGaP is stored in hundreds of files for each consent groups. For example, here we're looking at a small snapshot of the list of files for the Framingham Heart study. Next slide, please. So this type of organization puts the onus on the researcher to access all the files for each study and consent group, to decrypt the files as necessary, and then to merge the files as necessary for analysis. And especially when you're working with multiple studies and consent groups, this can be extremely challenging. Next slide, please. The PIC-SURE API ingests data from dbGaP and pulls out the information at the variable level. So everything is already combined for the researcher to run any analysis, and a user can search and retrieve data for any study or consent group they are authorized to access. This greatly simplifies the user journey. Next slide, please. Thank you. So now to walk through some use cases. The first use case I'm going to present highlights the interoperability between PIC-SURE and 7 bridges, which was used to reproduce the study using the COVID-19 ORCID data. Some of you might be familiar with this study, which was published in November 2020, titled Effective Hydroxychloroquine on Clinical Status at 14 Days in Hospitalized Patients with COVID-19, which concluded that hydroxychloroquine was not an effective treatment for COVID-19. We leveraged the interoperability between PIC-SURE and 7 bridges in Biodata Catalyst to successfully reproduce the results and analysis of this paper. Next slide, please. Thank you. So what do I mean by successfully reproducing the results and analysis, and what is the impact of this use case? We use the PIC-SURE API to extract the necessary ORCID patient-level data from dbGaP, as well as the data dictionary used for the study. We then use the PIC-SURE API to connect to 7bridges and directly import our selected data into the 7bridges analysis workspace. We piece together code from the original manuscript by contacting the original authors and by working with the limited code which was published online, and combined everything into a single R notebook, which is executable on 7bridges. We successfully created an R notebook where a user can, with one click, use the PIC-SURE API to extract the data of interest, export it to the analysis workspace, and then reproduce the exact analysis performed by this paper. So here's an example of the notebook itself. It is publicly available as part of our PIC-SURE examples, and anyone with the appropriate dbGaP authorization can access this notebook and reproduce this analysis in 7bridges right now. Next slide, please. On the top left of the slide, you can see an example of figure 2 from the original paper. And on the bottom right is our analog, the figure that we produced using PIC-SURE and 7bridges. You can see that they're virtually identical, and we have actually submitted a publication for this work demonstrating the reproducibility of the original paper. Next slide, thank you. Our second use case demonstrates interoperability between PIC-SURE and Terra with a focus on sickle cell disease research. Next slide, please. HCT for SCD stands for hematopoietic cell transplant for sickle cell disease. We collaborated with a sickle cell researcher to use PIC-SURE and Terra to conduct an analytic research study. This researcher was initially working within their own local environment and within the Sickle Cell Initiative web environment. We introduced them to Biodata Catalyst and were able to help them leverage the interoperability between PIC-SURE and Terra to first replicate their existing work and then build upon their existing analyses. We created a Jupyter notebook using the PIC-SURE API to build a cohort and perform analysis in Terra, which involved extracting the data dictionary, building queries to receive the data, testing reproducibility, and expanding the analysis. A manuscript containing this work is currently in preparation, and a portion of this code is publicly available on our GitHub as an example of how the PIC-SURE API can be used in sickle cell research. Next slide, please. Oh, sorry, you're already here. So here's an example of what the Jupyter notebook looks like in Terra. And in both of our use cases that I presented today, they demonstrate how tools within PIC-SURE allow us to curate data and then bring it into an analysis platform. These examples use 7bridges and Terra, but the PIC-SURE API is publicly available and scalable to additional platforms. So now I will go ahead and pass it over to Paul. Paul, I think you're muted or your mic's not working.
Hey, can you hear me now? Perfect.
Yeah.
Yep. So yes, thanks Simran. Simran showed you how the Biodata Catalyst PIC-SURE platform was leveraging different studies, and PIC-SURE as an open source project is being used in 11 other funded projects. One of them is the GIC network, and your one from NCAT, that enables to bring together patient-level data from now 5 different institutions and scaling to 9, where we have between a federated approach where the data stays within the various institutions, and then patient-level data or aggregate count, depending on the research project, can move to a central portal, enabling to query across all the different institutions. So showing you between, for example, Boston Children's Hospital, Cincinnati, and CHOP, where we have access to the number of patient samples across the institution with the various breakdown as it's being done with PIC-SURE. Taking this into account and looking at how this great conference went through with all the requests for search, definitely there's a PIC-SURE could be one of the options to be able to search across the different NCPI platforms by being able to query and extract any kind of clinical data, EHR, registry, clinical trials, connecting also with the FHIR API, any sequencing data, any biosamples, and any index files about radiology, MRI, EEG, any kind of files that can be indexed to know where those files are. So I'm going to show you 4 slides based on 4 potential ways of doing this. The first one, centralized approach, where we would have all patient-level data being into one central database. That's what we've been doing for Biodata Catalyst, but being able to do this across all the different platforms based on the various consent and authorization is unlikely to happen, but just as this is just a centralized approach is possible, but when this is not possible, there's the federated approach, as we are doing with the U1 GIC project, where the patient level data stays within each of the platform. There's picture being installed at each of the sites and enable using picture meta API to query using picture API across the different platforms to be able to query into one central place. Third solution would be the patient level data stays in each platform, and then the index of all the files are being centralized. So with no patient data, just the index that enable to search across the different elements. And the last one is because picture is a meta API. So by construction, it enables to augment and access all the different API, including itself. And then it enables to do aggregate data sharing resource or pass-through resource that enables to connect to other APIs. So another solution would be a meta API approach, leveraging the existing API from Envil, Terra, 7 Bridges, Gen3, Picture, all of us, Kids First, and 3C API that already exist that enable to have different search function that can be queried with this meta API that just pass through the existing information. So of course, this is just opens much more questions and discussion of how they could be done. But just when I saw the different discussions that were going on, those are potential things that we're definitely going to explore within the search user group and various resources on about picture and biodata catalyst powered by picture. And I really enjoyed those presentations today and yesterday. Thank you very much. Any questions?
Thank you, Paul and Simran. Excellent presentations. We have time for questions. Yeah.
So, Paul, it was interesting in the federated approach. Do you think it's possible to do a federation that has similar capabilities without installing some type of software on all of the federated pieces?
Yes. Either, well, there has, and that's on purpose, that's why I created those 4 slides. Either, first one, centralized, everything is put in one place. We know that this is unlikely to happen, but I just have to point it out. But this would be the easiest on a technical point of view, but the hardest on the policy point of view. Federated approach, there has to be something. So either there's picture being deployed across the platform. This is exactly what we're doing for the U1, creating the biggest pediatric network nationwide. Or if we don't want to use and install the picture API, we can, because picture is a meta API, and that's the option number 4, we can leverage the existing API to be able to connect to those different API. This is more work on the interface across, because there needs to be interaction that has to be done between the various API to the meta API. It's doable. We've done it with many different existing APIs, but it's just and it can be a mixed solution. There can be, it's not just one or the other. It really depends on what can be authorized. So this was just to throw out different technical solution that currently exist. We've done it on across many different projects. The last one, the meta API approach, we haven't done it with all those APIs yet, but that's definitely something we would love to explore. And another approach is the mixed approach, also by having the index being centralized. And that's what we are doing with the biodata catalyst right now.
Okay, I think I understand now that the federated and meta API approaches, while they have some equivalency, they're not totally equivalent. They're different. They have some different functionalities.
Correct, and different work on what do you install locally.
Right.
Yep. Perfect. Thank you.
but it can be mixed. It doesn't have to be one or the other. That's the most important. It's flexible.
Right.
Asiyah?
Yeah, so my question is about the status of this use case. So this use case has been existing before I came. So I do not have a one-pager for this, and it looks like it is already a mature NCPI use case. So, and there is a YouTube for it. So if it's a mature use case, how would we make it as a training material to be promoted, like to be disseminated?
On which one? Sorry, which use case you're talking about? The
yeah. So we need to go back to.
Yeah, I can speak to this a little bit. So I think our concern was as well. We're interoperating. Biodata Catalyst is a little bit different as an ecosystem, as a platform of platforms. So we are interoperating with Terra and 7 bridges within Biodata Catalyst. We want to take it the next step further to interoperate with Anvil or Kids First or another ecosystem. And that's not, we haven't got there yet. So I think that's the missing piece of the use case.
Okay, I got it. Yeah. Thank you.
I have a related question. So if in terms of implementation, although technically more challenging, it seems to me the Meta API approach is very appealing. Is there a reason for, I mean, to me, I'd love to see the Anvil trying it out with you. What do you think would be the impediment on actually really pursuing a more formal use case for that?
And it's really of who does the work. For example, between Picture Biodata Catalyst and Anvil, is it the Anvil team that look at how to integrate with the Picture API on their side? Or is it on the Picture team to receive the data and being able to exploit it? So there's all those 4 different use cases are possible. It's more of who does the work. And of course, in the context of a bigger vision, because right now, in many of those use cases that were presented throughout the days, there's a lot of one-on-one use cases. But as you were putting it and repeating, we want a much bigger vision across the whole platform. And so, do we have one perfect solution to do search and extraction of data across all the ecosystem that can work today? No, it's because it needs work and development from many different teams that have to work together. And this is just one option, one toolkit. And I've heard out 4 different ways that the word picture could be used, but there's also work from the other platforms. So it's more of making sure that we initiate more into a much more pragmatic approach now, because there's a more urgent need to make this happen. But definitely we are eager to collaborate on those topics.
Great. Okay, I think we ran out of time. I think that concludes the use case session. So what we have next on the agenda is Stan and John to summarize and synthesize. Thank you.
So I'm going to attempt to walk through what we've talked about yesterday and today and try to lay out what I've been able to see along with a bunch of extraordinarily capable folks that were helping about some possible use cases. Next steps. And in addition, I will then call on John, and we've asked him to perhaps MC a discussion among the NIHers here about what we should be thinking of as a group for the next 6 months or the next 12 months. So next page, please. So I think that we do have some fairly clear goals that are emerging as far as some of the technologies that are now getting to a mature state. And I think there's a common motif that's emerging that we need to, well, I'll step even back one more step. We're getting science done. I mean, this is clearly a dramatic improvement in the ecosystem since 2 years ago. So this is real progress along many fronts, and I'm really pleased, and it's been a joy to be with you all for these 2 days. There's another motif though that is emerging, not just success. Success is good, but we need to continue to focus our attention on user-centered activities, that is, use cases. We need to think about how we make this assemblages better, to improve the design so it's more user-friendly, and we still have some functionality issues that we need to work through, both in terms of ease of use, but also understanding the use so that we can start to do tests like we just saw of running things on different platforms and asking why are we getting variations. So at PFB, we need to identify and document the use cases that would result in the utility of PFB Lite versus PFB. I think we should take the energy, we should use time and energy to differentiate the utility more clearly between PFB, VDB, and FHIR. And when I say versus FHIR, I don't mean that these are in competition. I think they actually are different tools for different purposes, and we need to be able to say, here's why you would use one versus another. And then we ultimately need to clarify what PFB is and is not, and that is in the glossary, and we can continue to work on that and make some progress. Feel free to stop me, raise your hand anytime. I'm just going to try to get through these so we can hear from NIH. FHIR, here there's clearly a desire to aligning on a research study and metadata version 1 representation so that we have a clean way to represent data that we want to feed out in a more public fashion. We should try to identify, and I know all of this work is going on already in the FHIR community, roadmaps for the platforms to use as we understand how things are going to be developing in the FHIR community and new tools roll out, and understand the services that will be available, the use cases that might be supported, and what limitations there are. And then obviously we need to continue work on the existing FHIR use cases, which, by the way, we need to document better, because now that Asiyah has done such a wonderful job of putting this up in GitHub, we have a much more organized way of talking about what NCPI as a group is doing. RAS, the current plan has to be completed. Brian was very clear about the necessity of trying to stay as close as possible to the timeline that's been established. I know that's challenging in some cases, but we really have to do that. So solve the challenges of milestone 3, which includes single sign-ons, and we need to meet the deadline. And then we need to have some discussions about what we need to do beyond the milestone 3. What are the next steps, and how do we extend the passport, in some cases perhaps the passport language, that will allow us to both parse and translate between different passport systems, particularly those arising from overseas potentially. Next page, please. And if you disagree with anything, don't hesitate to yell. The end user cloud cost, that's clearly something that is really a high priority. We have to figure out how to attack this problem. And one very creative solution that came out was to create free workspaces for training in the cloud and let people start to experiment in this space. Also, we need some budget templates and guides so that people can at least estimate. I know it's not possible to do this with a high degree of accuracy at this time, but it's still important. End-to-end user stories that have resulted from people using the system and understanding how much it costs is essential. So we really need to create a database of cost modeling efforts across NCPI. And in the long term, perhaps we need to carefully think through how we could get together collectively and do some kind of code-a-thon and try to experiment in this space collectively, and also include some of our students and other people who are interested in starting to use the platforms that we're developing in order to understand cloud costs better. And that would be very important for many many different end user groups, including universities that are now going to the cloud to teach classes, which is happening much faster than I anticipated it would. Search, we have to make sure that we stay focused on how the users are thinking about using search, and there are not a consistent set of uses, so we're probably going to need to see multiple types of search implemented. I think that's emerging in lots of different forums. So there's a desire to form a working group that'll drive the development of use cases so that we understand the different pros and cons of using different search techniques, so we can develop personas, we can make guides to existing searches and components, and ultimately be able to help users make informed use of the tools that we're building. We also probably ought to create a list of search components so that we can plug and play different pieces. I think that's something that we could do as a community, put some of the pieces in the open source, and also documentation so that we can start to reconfigure search using the collective pieces that we've built. And I think it's a fantastic idea to try to create a search taxonomy so that we can inform both a research and a search roadmap. That's not probably terribly easy, but I think it's something really worth spending some time on. And then everybody knows that there's a search RFI on the street. I don't know if Susan is still with us on the phone call, but we probably need to collectively respond to that, or at least as small groups, make sure that we have our input, because the workshop that will be held is likely to have an impact on where the search activities, research activities in search go in the future. And then defining and promoting semantic maturity in the data is clearly a high priority. And by semantic maturity, we don't necessarily mean, although perhaps we in some cases do mean, adding a whole lot more semantic information, metadata, but at least making sure that we get the right amount in so that we can enable fundamental utility. Next page, please. So other interoperability, really interesting stuff going on in that space, engaging users for testing and current functionality, and then gathering some feedback on new features. Now that we're seeing a lot of people, I remember, I think it was as recent as a year ago, we weren't exactly sure who we could talk to who would have a good use case on using multiple platforms. And now Melissa has not only talked about it, but she's done a good portion of it. So that's fantastic. Development of use cases to drive interop features so that we can think through the potential of different ways to approach putting together the tools or functions that are appearing on each of the platforms. We have to find a way to standardize tools and apps and the deployment, which includes doing testing for securities and trust. And then finally, developing methods, methodologies, processes to publish completed use cases so that we can both replicate those and train on it. And in fact, we need to go ahead and develop training on interoperability methods. GA4GH, they're constantly developing new standards. We can participate by getting engaged and coordinating our representation. We have a number of people in our group that regularly attend, so we should be cognizant of the fact that we want to make sure that we lodge some of our interest into those representatives so that it gets taken up by the GA4GH community. We should document the standards that are in use across NCPI and think carefully through about where we want to go in trying to help shape GA4GH's activities in the future. And then think about creating some new standards or proposing some new standards to propose into GA4GH. They will have a new pedigree ontology coming up fairly soon, and that implementation on FHIR is going to be done in, what, a couple of days, a week? Yeah, about a week. Okay, next page. Very interesting discussion of use cases. Thank you all for arranging that. The structure is clearly now in place with Asiyah's work in setting things up so that we can coordinate and have a much greater degree of transparency. And that will in turn drive and extend the utility of these use cases. And it's incredibly exciting to see both many mature use cases that are yielding real science and also new use cases that are being developed to drive the activities of us jointly together. It was very interesting to see DERS being used as through a distributed pipeline to understand sex as a biological variable. I think that work is absolutely fascinating. Also very interesting to see the complementarity of DERS and FIRE and when you might be able to use both. I really thought the comparison that Owen talked about, comparing the algorithms across platforms to look at the results, was very interesting, and we need to continue work to both ascertain the reasons behind the differences in the results, but it may be that we find that the differences in results are for good reasons, and if that's the case, we'll need to think through how we, I'll call it message, but explain the fact that you're going to get differences under certain circumstances. I don't know if that's true or not, but it does seem plausible, at least given the stochastic nature of some of the approaches. And then the meta API approach that might be adoptable, that's what Paul talked about, cross NCPI, I thought was fascinating. And we probably need to develop a use case that we can use to drive that work and test it. And then clearly there's some work on trying to decide on, from a governance standpoint, where the development gets done. So I'll end, I think this is the last slide that I was going to talk to, and then I'll turn it over to John to orchestrate some discussion. I think it was really important that multiple times we pointed out to each other that this is a cultural change as well as dramatic technical changes, so it's not easy, and it isn't always received naturally, received in a way that matches the speed that we wish it would be, but we're clearly making progress in putting together systems that can interoperate and make a difference in how well we can do science and how well we can train the next cadre of scientists. So we need to find opportunities to leverage the program resources that we're putting in play across these different centers and institutes to maximize the impact, and so that the importance of what we're doing is both visible and obvious. And we have a lot of utility now that we can deploy, but in some cases it takes an expert to do it, and we sure need to look for places where we have the possibility of an easy button. So we can take some time to talk about the topics on each of these slides, and I urge you to correct either directly to me, or you can go into the slides themselves and make edits if there are errors on these slides, because these are interpretations from a group of us and we may not have gotten it right. So do we want to discuss for a few minutes? We have about 20 minutes left before we're out of time. John, do you have a lot to say here, or do you have a lot to orchestrate?
We are going to sort of review a little bit about what we talked about in our NIH only session, and then kind of open up for comments. So if we want to have a few questions or comments now, that's fine.
So any questions and comments about this summary and next steps? I'm sure we're going to go back and look at this more closely and make sure that we try to synthesize to a greater degree where possible. So if we missed any major things, I really would like to know about it. Well, John, I think you're up.
All right. I think it's getting towards the end of the day, so folks are maybe a little talked out. But before we get going, I want to just really thank Stan and Becky and Sarah and the whole group at RENCI and RTI for setting up this 2 days of discussions. I think it was a great meeting. I really liked the focus on sort of these different work streams, and I think we have some good actionable next steps in each of these areas that hopefully can push the ball down the road a little bit. I also think there is some growing level of maturity of the NCPI group, which I think is evident in what we're seeing in the use cases as well as in our discussions, and I think with Susan and ODSS kind of really leaning in here, I think it's poised to really help accelerate some of that activity. So I think this is an exciting time for the NCPI group. So again, thanks to everyone for participating. So we had a NIH discussion a little while ago, and I'm not seeing oh, there she is. Valentina was going to sort of lead us off a little bit on a recap of what we talked about.
Yeah, thank you, John. Very briefly, I also want to thank RANSI and NHLBI for hosting this meeting, organizing it. It went beautifully, and I think we're all happy with the outcome of this meeting. And some of the things that we discussed in the NIH internal group earlier on basically are similar to the themes that we heard in Stan's summary. So anyway, I also want to give time to the people who raised some of these topics to also speak up. So let me start first of all with something that Chris Kinsinger and Jamie suggested. Chris, do you want to?
Yeah, sure. So I'm Chris Kinsinger, and I represent the Common Fund Data Ecosystem at NIH. And one thing we're thinking, is it within the scope of NCPI, as you're developing best practices and standards and as they mature, does it make sense for NCPI to actually provide a service where as others begin to implement those best practices and service and standards, that you would, someone from NCPI could check the implementation to make sure it's done right? And maybe that would start with like, here's a package of code that's a test case, and you could see if that test case runs successfully. And then as it gets more complicated, maybe there would be people who would actually come alongside and help. But just, you know, as these things are maturing, which it sounds like they are, would that fit within the scope? And specifically within the Common Fund Data Ecosystem, we have about 4 or 5 data coordinating centers that are looking to implement DERS in the coming year, and we're pretty excited about that. But we're working fairly closely with Kids First. But you know, beyond that, how do we test that we have interoperability working with some of the other platforms represented here? So just wanted to throw that out there in the mix and see if it sticks anywhere.
Yeah, and what came up, I think, is completely in line with what is the stated mission of NCPI, which is really to develop best practices, you know, tools that can be redeployed and easily used across NIH for related projects and activities. So that seemed to be a very nice suggestion that we could think about pursuing. Right. So the next thing that I also wanted to highlight is that, of course, we talked about RAS and how important it is to reiterate that it clearly is a major bottleneck, is something that we need to fix as soon as possible. We have some timelines, and hopefully we're going to be on top of it and deliver as planned. We had discussed about how to ensure that we're going to check on progress and so on. I do not know if Alistair is on, or Rebecca, if they want to add anything.
Yeah, you know, one of the things we talked about is how we make sure that we sort of maintain focus across all of the platforms, and that, you know, there are multiple efforts going on in this that all have to be coordinated, and that really we need regular touch points. And we talked about ODSS perhaps convening kind of a group to provide that sort of regular touch point to make sure the communication is flowing the way that it needs to, and that everyone understands what's going on. You know, we've sort of observed at times that there's been a little bit of disconnect between the teams as to who is supposed to be doing what, and we can't afford to have those happen. Given where we are, there are so many time critical things. And so you should see some invitations for some kind of regular touch base, whether it's a weekly scrum or whatever, just to make sure that we're tracking and keeping things moving.
Great, that's great. Okay. So we discussed briefly the establishment of the search working group. There is some concern about adding another working group and how that is going to have an impact on, the number of people and staff that is already involved in this large number of NCPI related activities. So that is clearly a concern. At the same time, we also understand that doing search is showcasing the ability to do searches across NCPI platforms is important. I'm not sure if Asiyah wants to say more about this.
Yeah, I think the Stan has his summary slide laid out the discussion point very nicely and aligned with what we just discussed. So to move forward, it seems like search is a need from the community, and we need to figure out what kind of group we will form and what are the people who will take the lead. And then another rising thing I think was I just feel like needed to add to that list is the tools comparison. Like Paul just presented PIC-SURE, and I think Gen 3 has also search capacity and FIRE as well. This might be another task of the working group, and then the priority needs to be set. I really think define the persona and collectively respond to the RFI that ODSS put outside will be an immediate priority for this group, but we have to form the group first.
Yeah. And finally, one last topic that came up is about training, especially there were two things that resonated in particular in our group. One is how to educate students and administrators. And the other one is exploring this possibility of making available some free space for cloud use for students. So James, Asiyah, do you want to add anything to it? I don't see James.
Yeah. I can start. So I just feel like a lot of people comment about the this NCPI community to be more integrated or extended or interact with the more global, bigger picture, bigger group. So especially with training aspect, we might be able to collaborate with some bigger vendors. So that is the idea put out there on the table.
Oh, I see James also online.
Yeah, so I'm a former lab person, and I ran a lab for years and trained graduate students, and I'm really interested in the training kind of aspects and making transition to the cloud resources really easier for developmental biologists, my field, but biologists in general. And so the ideas that came out in terms of making cloud space available so you could have that graduate student or undergraduate who's rotating in your lab available, I think was a really wonderful suggestion. And we'll see on this end of things what we can do, but we do have the opportunity to increase training, and that's going to be a really important aspect, I think, of not only the NCPI efforts, but across NIH, because we don't want to reinvent the wheel. We want to coordinate with other groups trying to do the same thing. It's been recognized widely across NIH that this is a need. So anyway, I'm delighted that the difference between where we were 2 years ago and where we are now seems to be really apparent in this group. And I think this is a great effort, and it's really important that you all from the external community is involved in it. That's all.
Great, thank you. Does anyone of my colleagues want to add anything? Did I forget anything? John, is all yours.
Any responses or questions from the broader group?
I guess this is Valerie. One question I have is, especially with the formation of this search working group, I mean, Asiyah did a really nice job presenting about our sort of use cases first approach with the way we're going to organize and the way we're going to drive the development work moving forward. And Stan, I think your slides really beautifully summarize this. So I just wonder, with the establishment of a potential search working group, if we should actually start from a place of getting some potential use cases documented and at least assess those in the broader group before we start scheduling meetings, but it's just a thought.
Good, Stan.
So that I just wanted to respond to that. I think we should adopt a principle, and we should write into any working group or tiger group that we choose to stand up, that we start with use cases, defining the use cases, and that to some degree should take some of the anxiety about who's going to win, just to put it bluntly, out of the picture, because there's good use cases should permit different technologies to shine. And we want to be able to demonstrate the fact that there are completely legitimate differences. This is a very hard problem that's been studied for many years. So that's, I think, maybe an operating principle that we don't have any governance for NCPI, but we could say from now on, this is the way we're going to operate.
Yeah, I think that kind of is the governance, for now, that everything's anchored to use cases. Yep. So that's pretty good.
Go ahead, Becky.
Yeah, I was just going to say, I think Stan and I spent some time reflecting over our last in person and thinking about where we've come. And I feel like the story to date has largely been success interoperating around technologies, largely APIs and standards adoption across the platforms. And I feel like one of the themes that I didn't see coming, but I'm excited by coming out of this meeting, is that, yeah, we're doing that, but now there's an opportunity to kind of collect resources on overlapping activities like training and GA4GH representation and these things that are common across all our programs, and see if we can make those like more impactful or efficient by virtue of, I don't know, sort of our collective size, I suppose.
Yeah, I agree. And I think that's going to be key to all of our successes, in the sense that there is a lot of overlap in what we do, not only in terms of education and resources, but quite frankly, in the services that we offer. And so, I think for being able to sort of, as you say, magnify it, but also, from a sustainability standpoint, from a, financial standpoint, for a lack of a more subtle term, we need to be able to leverage that because, there is a lot of pressure on the NIH budget right now, and we need to figure out ways where we can continue to innovate, but then also innovate and expand our user base, but then also conserve resources in other places. And so this may be one of the strengths of this group, is to figure out where some of that resourcefulness can come from. Stan, you had your hand up.
John, I apologize before speaking. This may not be necessarily very politic. So I hear what you're saying about resources. I totally get it, totally understand. Many of us have been doing this NCPI stuff as an addendum to the work that we originally took on. There's been some funding for various pieces, so I'm not complaining in any way. But the fact that we've made this much progress, in my opinion, is almost miraculous. Now, maybe I'm praising here, but it's still an incredible team science effort to take groups working from completely different funding streams and manage to get interesting science out the door, in addition to trying to build a very complex, technologically challenging collection of platforms. So the non-politic part is, what are you all going to do to make sure that this gets transmitted up to leadership, particularly leadership that, you know, in some cases is in transition. You would not believe how much we all were provided with anxiety hearing that Dr. Collins is going to be leaving eventually, because he's been a big proponent of this. He's come and talked to almost all, you know, many of our meetings. So can we ask you all we got to work at this together, if you get my drift.
Yeah, so I'll take a stab. I mean, this is a big question, but I'll take one small stab at it, and then see if others can chime in. So, you know, a couple things. I do think that you know, the use case science-focused aspect of this is very helpful, because nothing speaks to NIH leadership like the generation of knowledge. And if we are promoting, you know, discovery in a way that was not possible beforehand through, you know, scale or speed or, you know, variety of data that's brought together, then, you know, that's a tremendous selling point for our system, or our system of systems, if you will. You know, Mike Fiello messaged me. I'm hoping I'm not saying anything that he won't share publicly, but, you know, he talked about sort of some standardized use metrics that, you know, we probably need to think a little bit about, so that we can show that we are in fact growing a user base here. You know, there was a lot of interesting talk about, you know, the cultural barriers to getting into the cloud, and more than cultural, but there's financial and logistical and regulatory. You know, we're not going to be able to grow a user base unless we can sort of address some of those things. So trying to figure out how to do that, I think, is going to be critical, so that we can show that we are slowly but steadily bringing in a wider and wider circle of people. And actually that brings me to one small point, but which I raised in the comments a while ago when talking about search and search use cases. Someone said, well, you know, it's hard to find users. We can use the BDC fellows, the Biodata Catalyst fellows, which sure we can, but that's not expanding our circle, right? Those folks actually know how to navigate the system pretty well. We need to find the people who are having trouble figuring out where the data is, understanding what their needs are, and then speaking to those needs through our technology. So I think, you know, showing growth in metrics that speak to leadership, I think showing that there's science getting done that couldn't get done otherwise, I think are two, you know, significant selling points. And I think, you know, speaking to our internal leadership and making sure they're speaking to the new NIH leadership when they come by, to make sure, when they come on, to make sure that they see, like Dr. Collins did, the value of this, I think is going to be important. I saw Mike had his hand up, but then he took it down. Mike, did you want to say something?
Yeah, I just wanted to say that, you know, I messaged you that existential justification is needed by leadership, obviously, and that's going to take the form of use metrics, unfortunately, to some degree. So we have to be prepared for that and have an answer for that. The other thing that I see as a potential thing that wasn't brought up but probably should be is that we're going to need experts that are not necessarily from each platform, but knowledgeable about cross-platform use. and not beholden to one platform, because we don't want to set up that kind of competition. We need, I don't know if it's fellows or I don't know what it is, but that could be something that NIH can, we could suggest NIH fund some, and I don't want to call it a help desk, but it would be, trainers essentially, because a lot of people are not using the cloud right now. A lot of people are, but a lot of the dbGaP users I know have plenty of space on their academic clusters, and they're happy with downloading the data, even if it takes 2 weeks to run their analyses. So anyway, those are my comments.
Great. James, we'll give you the very last word since we're 2 minutes over.
What a responsibility. I just wanted to say that, regardless of whoever Dr. Collins' successor is, the value of large data efforts, especially around genomics and, well, all of these large data efforts, is pretty deeply ingrained now into NIH Institute leadership, at least certainly at NICHD. There's been a lot of verbiage, a lot of talking, a lot of selling done here by NIH staff, and it's not going to go back. We do need to have metrics and scientific advances that amplify what our message has been, but there shouldn't be any anxiety that the new director is going to come in and sweep away these kind of efforts. They're long-term, they're long-term, but in many fields, there's no other way for rapid advancement, and that's really clear, that we can make rapid movement in scientific knowledge and hopefully impact the health of the nation by improving it through large data efforts. And you guys are tip of the spear here, and that's really wonderful. That's all.
All right, I'll hand it back to the RENCI folks to get us out of here.
Let's go to don't forget the NCPI glossary. We'll send it out again just to remind everybody. And next slide, and we'll get this done quickly. Thank you for attending. There is a workshop evaluation form. If you like this format, tell us. If you don't like it, tell us. We're going to be back in spring. I'm not sure who the next host is. I can't remember.
Probably going to be us, the Anvil.
Anvil. Then Anvil will get the benefit of the feedback. Thank you all.
Bye.
All right, thank you, everyone.
Thank you.
Thanks so much.
Hi, thank you. Yeah, great.